Well, we have a really exciting episode tonight. I've been looking forward to doing this. It took a little while because it is about one of my favorite topics, genomics. And when we think about genomics, there are some controversial components like stem cells, cloning, and precision medicine. So this is going to be exciting. It's going to be fun-filled. And also we're going to talk about some fun things like Jurassic Park, a real-life Jurassic Park. So stay tuned, ladies and gentlemen, for another episode of Topics Discuss. Welcome. Interesting topic tonight, folks, ladies and gentlemen. So what comes to mind when we think about genomics, or when you think about genomics? Do you think about genetically modified food or GMOs? Do you think about cloning dinosaurs to create a Jurassic Park? Cloning and editing superhumans to create a super army? Or do you think about the controversy associated with embryonic stem cell research? Are we playing God? Or are we just discovering the powers of God that exist in each of us as human beings? Let's get into it. So genetics and genomics both play roles in health and disease. So when we think about the difference between genetics and genomics, it is important to remember that genetics refer to the understanding of our genes in terms of the way that traits are passed down from one generation to another. Genomics, on the other hand, describes all of a person's genes. We'll get into what that means here as we move through this podcast. But I want to start first with talking about what the word gene means. What do we mean by gene? So when thinking about our genetics or our inherited traits from our parents, the term gene can indicate a unit of heredity which is transferred from a parent to offspring and is held to determine a unit of heredity or a characteristic for the child and offspring. But when thinking in terms of genomics, I like to use a more technical term or discuss genomics more around understanding a gene and in a distinct way, as a distinct sequence of nucleotides forming part of a chromosome. So one of the most basic things or most basic ways to potentially describe a gene and how genomics impacts our daily lives is to liken the genomic code to a binary computer program. So when you think about a computer program, you can think about it in terms of ones and zeros. And the ones and zeros are used to create a program, and the program then tells the computer how to operate. It's basically software programming, right? And you have program start, and stop codons or codes. Well, you have the same thing. This is way oversimplifying, but you kind of have the same thing with the human genome. Except in the case of the human genome, the computer code is made up of a combination of 4 amino acids. Rather than ones and zeros, you have a combination or a codex of A, G, T, C, or adenine, thymine, guanine, and cytosine. Those are amino acids that when combined, they will create proteins. Not the kind of proteins that, don't think about this in terms of like your muscles and growing muscles throughout working out, but rather the proteins are really a chemical signal that creates a signaling pathway for the rest of your body in terms of how to operate. So that is the body's operating system. And you can think about our bodies as the hardware and our genome as the software. So let's use the human colon for an example in terms of how this works. The colon replicates the lining of its own lining, the epithelial cells, every 5 to 7 days. So how does that happen? Well, it happens because there are gene signals that come from our cells. Every single human cell has the entire genomic code encoded into them. So if a cell is differentiated to function like a colon cell, which in this case, in our example, it is, the colon cell will have the right code to turn on the mechanism to shed or kill off the old epithelial lining. And then there is another codex or codon or sequence that signals the cells of the colon to grow and to replace itself. So imagine if one of those codes was turned on in a hyper state, like replicating over and over again, or if the code didn't work to turn off or to shed the old epithelial lining. If you have mutations like that, then you can imagine that the cells of the colon, the lining of the colon, would begin to overly grow or overly proliferate, which really results in cancer. And that's all that cancer actually is. All kinds of cancer come from this genetic anomaly, or genetic mutation. And I've had some people say, there may be people that will comment that will try to say that genomics is not, or cancer is not a genetic disease or a genomic disease. And I'm telling you, it is absolutely related to our genomic code. And many diseases are, and we'll learn about that in our podcast today. So genetics, again, thinking about genetics, the researchers and scientists who are working on genetics really are concerning themselves with scientific studies of genes and their effects and how those are inherited, or even disorders are inherited from one generation to the other. So an example of this is cystic fibrosis, and I want to take some time to really talk about cystic fibrosis. It's a really good case study to understand genetics, genomics, and inherited disease. So it's worth spending some time on. So mutations in a gene called the CFTR, or the cystic fibrosis transmembrane regulator gene, the CFTR gene, causes cystic fibrosis. And that gene was actually discovered in 1989. In normal cells, the CFTR protein acts as a channel that allows cells to release chloride and other ions. But in people with cystic fibrosis, this protein is defective, and the cells do not release the chloride. So again, going back to the computer code, the cells have the right encoded or encrypted information to send the chemical signals to make the body function the way it should, like releasing chloride and other ions. So if there's a mutation in that gene, that channel is not going to work properly, and it will result in a disease like cystic fibrosis. In cystic fibrosis, the CFTR, that mutation causing this problem or erroneous signal, it results in improper salt balance in the cells. It also results in thick and sticky mucus generated in different parts of the endocrine system and in the lungs. So researchers are focused on ways to cure cystic fibrosis through genomics and actually correcting the defective gene, and therefore repairing the defective protein, because the gene creates a protein that then creates the signaling pathway. So there are gene therapies that have been utilized for some time to treat cystic fibrosis in patients. because it targets the cause of cystic fibrosis rather than just treating the symptoms. So gene therapy for cystic fibrosis started in 1990 when scientists successfully corrected the faulty CFTR gene by adding normal copies of the gene to laboratory cell cultures. Now, this is an important concept for the rest of the podcast because this methodology has really grown and developed over time. Now that we're in the 21st century, we can do so much more than what we're talking about here with cystic fibrosis. So by finding the best delivery system for transporting normal CFTR genes into the patient through synthetic vectors, nose drops, drizzling cells into the lungs, that will help to correct the mutation in the CFTR of that patient. So that methodology has worked, but as we move forward into the 21st century, how would gene editing help with cystic fibrosis? So as you think again about a gene functioning like a computer program with ones and zeros, but instead A, G, T, and C, editing a gene and providing an alternative pathway or a new computer program will actually, there's a way to access the cell's own repair mechanism, DNA repair mechanism, to utilize a new program that we give to the patient, or we inject, or we expose the cell to. Then the natural DNA repair mechanism can correct the mutation in the cell. So we'll talk about the different types of gene editing, and this is probably my most favorite part of genomics and genetics and thinking about all the possibilities, and it relates to CRISPR. So there are different editing tools that can be used to edit our genes, but CRISPR's probably the most versatile. In fact, you can buy home CRISPR kits, take them home, and work on hybridizing plants, if you will, or doing other experiments. So CRISPR gene editing includes a guide that locates the mutated sequence in the CTFR gene, and then a template with the correct segment of DNA. So if you think about introducing a new set of instructions, really, and then CRISPR has these scissors, if you will, that break the patient's DNA at the site of mutation where the mutation is found. And that's so important because it creates this precise mechanism to edit the gene, to remove the mutation, and then to introduce a repaired or normal gene sequence or a set of instructions. So once the tool enters the cell and reaches the mutated sequence of DNA, the scissors of CRISPR crisp out or scissor out or cut out the mutation. And then what that action actually, snipping the mutation out of DNA, attracts the attention of the cell's DNA repair mechanism. So it's almost like it induces a type of immune response. And the DNA repair mechanism gets into work and uses the template that's been introduced, the template with normal DNA, and uses that to fix the broken DNA that we've cut out. So in a way, CRISPR uses the body's own repair mechanism to introduce the new sequence, the new code. And that's pretty remarkable. And that will lead us down some pretty interesting ideas. So a major advantaging of gene editing is that the changes it makes to the DNA are permanent, because your own DNA repair mechanisms have made the change permanent in the cell. So every time the cell replicates, it's permanent, that change. So in practice, though, gene editing is not perfect. So I don't want to leave our audience thinking that that CRISPR is the solution to all of our illnesses and aging. But it is important to understand what the potentials are in the future. So gene editing right now is very active in terms of research nearing clinical trials, especially for cystic fibrosis and other diseases like sickle cell anemia and sickle cell disease. So I'm introducing to you CRISPR and genetics. Now let's talk a little bit more about what we mean by genomics and what is the difference between genomics and genetics. So genomics is a more recent term that's used today to describe all of a person's gene or all of a person's genome, not just the inherited portion or those traits linked to our parents, but the entire genome, some of which we don't really even fully understand today. So it's a scientific, it's complex scientifically, and it helps us to understand genomic-related diseases, such as heart disease, asthma, diabetes, and cancer, because these are diseases that aren't just environmentally related, but they also relate to there's a combination of impact with environmental factors, individual genes. So genomics is offering new possibilities of therapies and treatments for some of the most complex diseases. One other concept to introduce is something called population genomics. So it actually applies biotechnology in the genome in a way that we can characterize genetic variations and work toward understanding how they relate to different diseases and how they contribute generally to the health and well-being of people. And it will also help us to understand human evolutionary history and human evolutionary timelines. So the population study, genomic study I'm most familiar with, obviously, is hereditary. And I'll put links to these in the description below. But hereditary is a population genomic study with the goal of making new connections between gene and human disease, developing new treatments, and increasing our ability to predict and prevent serious disease now and in the future. So there is a whole nother podcast we could spend on hereditary and population genomics, but essentially what that allows us to do is to test a large population, 500,000 to 1 million voluntary participants who will voluntarily give their blood, and then from that blood voluntarily allow us to extract DNA from their blood, do a whole genome sequence, and then attach or associate or link the whole genome that we've conducted the study from sequencing associated with the patient's medical record. Again, all voluntary. So imagine if you had a population of 500,000 or 1 million individuals that participate in a study like this. You could then associate a whole genome, which includes gene mutations we don't really even understand now. We can take those and all of the medical records associated with those patients, and that could reveal genes that are associated with depression or anxiety, because we have the associated medical record. So just to give you a little bit of an explanation, a genotype is the whole genome, the whole code within each of our cells. But how that code expresses itself externally in our traits is called a phenotype. So the medical record is essentially our phenotype. It's how our genome is expressing itself, both in terms of our eye color and skin color and makeup, our body frame, and diseases that may express themselves through gene mutations that we can discover. So it's a really important endeavor that many institutions are working toward. So moving on to another part of the topic in terms of genetics. So why are genetics and genomics important to our health? And we just talked about all of the different components of that. So I'll just give you some additional examples. Genetics helps individuals and families learn about how conditions like sickle cell or cystic fibrosis are inherited in families that then will result in the need for screening and testing options and other preventive measures. So as we think about genomics, however, in helping researchers discover how our bodies interact with the environment, because the whole genome might be involved. So why do some people, for example, get sick with certain infections, environmental factors, and behaviors, while others do not? For example, there are some people who exercise their whole lives, eat a healthy diet, have regular medical checkups, and die of a heart attack at age 40. Why does that happen? Well, it's genomics. It's genetically related. There are also people who smoke, they're obese, they never exercise, they eat Kit Kats and peanut M&Ms and Twinkies every day, and then they live to be 100. And that doesn't feel very fair for those of us who try to live healthily. Why does that happen? Well, there's a genomic component. An example of this is COVID-19. So with the pandemic, some individuals became very ill and even died from COVID, but others didn't. Why is that? It's because of our genome. So our genomes actually indicate or can predict how we're going to respond to the COVID virus, for example, and other diseases. So I just want to be really clear about how much genomics relates to our health and how intertwined they are. So apart from accidents, here's some data, such as falls, motor vehicle accidents, or poisoning, genomic factors play a role in 9 of the 10 leading causes of death in the United States. So genomic diseases and genomic factors play a role in 9 of the 10 leading causes of death in the United States. So that's heart disease, diabetes, cancer. Isn't that interesting and so important? Yes, lifestyle is important. Yes, eating healthy and exercising is important. A really big portion of this, though, that cannot be understated is also our genomes. So all human beings, all of us, are 99.9% identical. So there's just the small 0.1 or 1/10% that hold important clues between genes and the environment by means of genomics that will help us, that influence the way that we can prevent disease and modify lifestyle in combination with genomics to detect early and prevent disease. So just as I kind of look through my notes and to try to make, there's so much to talk about in terms of genetics and genomics. I'm going to skip to a concept called Pharmacogenetics. So pharmacogenetics really is a field of study dealing with the variability of responses to medications due to variation in a single gene. So here's another example. Some patients who have depression or anxiety, and it's not resolvable through therapy and other mechanisms. So for their mental health issues, medication may be required. But many patients will experience taking a antidepressant, an SSRI or SRDI or whatever mental health medication their provider has prescribed them, and they have they don't react well to it, or they don't feel better, or they feel worse. And sometimes that is a result of genetic variations in each of us individually that determines how we're going to metabolize and process and transport a drug. And so some institutions who are using pharmacogenomics with their patients are finding that physicians, providers who start with pharmacogenomic testing first to understand what's happening in a patient's genome, to help guide them in the type of antidepressant and the dosage, typically will see a better response initially. Because if you think about it, if you are having mental health challenges and problems, and you have exhausted all your options, you're ready for an antidepressant or some other mental health medication, and then you try it and it makes you worse, then not only do those patients oftentimes never want to try a medication again, which could really save their lives, but they may not even want to go back to therapy. They may not want to see their physician who prescribed it. It confirms all of their fears about antidepressants. So pharmacogenomics becomes an extremely important part of helping providers better guide their treatment for patients. Now, this is not just, pharmacogenomics just is not related to only antidepressants. It's also related to a whole bunch of different drugs and how drugs interact with genes. So it's a really important future for precision medicine and how genomic medicine impacts our lives. So now we get into some even more tantalizing components of genomics, and it's stem cell therapy. So hang on to your hats for this next section. Stem cells have two important characteristics. First, stem cells are unspecialized cells that can develop into various specialized body cells. So if you think about an embryo, when an egg and sperm come together and fertilize, the first 6 to 7 days or so of that fertilization creates something called a blastocyte. And blastocytes are not really embryos yet, but they're a clump of stem cells. That clump of stem cells, however, has every stem cell is the incomplete genomic code for that new life that has been created. And what begins to happen over time is that those cells with the code in each of them begin to differentiate and specialize and become cells like muscle cells, neuronal cells, skin cells, blood cells. And so as that happens and we are formed as humans, as adults, we still have some stem cells inside of us. Our liver, for example, our bone marrow. But those stem cells are kind of like prepared to become the organ that they're associated with. So they're kind of differentiated already. So a liver stem cell, for example, is really only prepared to differentiate further into a liver cell, and that is a reparative mechanism that our bodies have. Sometimes people will go to Mexico or some place and they think they're getting stem cells, and it's unfortunate because people think that they're getting this embryonic stem cell that can differentiate into anything magically, into any tissue that needs to be repaired. When you don't, first of all, you don't know what a provider in some, I don't know, dentist office or chiropractor's office that's claiming they have stem cells those stem cells might come from the umbilical cords of cows. And if you think about that, that means this person that's injecting stem cells into you is injecting a foreign agent. The stem cells in an umbilical cord of a cow are already differentiated to become cows, not human tissue. And in fact, our bodies would see those stem cells as foreign, and even reject it and process it through macrophages and just get rid of it. Now, there are some family members of mine who are going to watch this and they're going to dismiss what I'm saying, and they're going to say, well, Gary's not a doctor. What does he know? You're right. I'm not a doctor. And what do I know? So I guess you can figure out on your own what stem cell therapy is really all about. But I can tell you what the data tells us, right? So with stem cells, one of the controversial components of having stem cells is, or using stem cells for treatment, is the most effective type of stem cell is an embryonic stem cell, right when the sperm and an egg come together and create that blastocyte, those initial first stem cells. Those stem cells can differentiate into any organ, any human organ. There are two problems with that. First of all, there's ethical considerations regarding, creating, using an egg and a sperm to create this blastocyte that you're going to then use for medical research or medical treatment. But secondarily, using embryonic stem cells to treat disease in another human will have some of the problems that we've talked about in terms of rejection. Just like an organ transplant that is coming from another human, it's not your own tissue, so there's always a risk for rejection. So one of the cool things that has happened with CRISPR is the ability to take differentiated cells from your adult body. So if skin cells were taken from my body, for example, CRISPR can be used to reprogram that skin cell and knock out the genes that told these cells to produce skin, told these cells to become skin cells. So CRISPR can be used to knock those genes out, take them away so that there is no gene telling those skin cells to turn into skin, or telling those stem cells to turn into skin cells. So I'm oversimplifying, but CRISPR knocks those genes out with instruction for becoming differentiated, which then results in that adult skin cell becoming kind of like an embryonic stem cell. It reverts back to its stem cell state. But here's what's cool. That is your own stem cell. So it's coming from you. It is not going to be rejected if we used it in treating a disease. So let's imagine that you are in a bike accident. Some of my viewers love to bike, and I'm always worried they're going to get hit by a car because that happens frequently. So be careful. But let's say that happens and your spinal cord is severed or severely damaged. Then theoretically, with this new technology, blood, skin tissue, or even bone marrow, some cells from your body can be extracted. The encoding genes that tell those cells how to differentiate can be knocked out by CRISPR, resulting in a clump of new stem cells that belong to you. And what's awesome and fascinating about these engineered stem cells is that they are not embryonic. So they have not come from a sperm and an egg from a donor. They come from you as the donor and your own cells, not even a reproductive cell, but your own somatic cells. And then they also have the ability to replicate and divide and replicate and divide and replicate and grow forever. So they're they're eternal and immortal, which is fascinating to me. And so if those induced stem cells that we've induced through CRISPR are inserted into the damaged part of the spinal cord, those stem cells will then begin to differentiate naturally into new neuronal cells to repair the spinal cord. Is that fascinating or what? Now, there have to be clinical trials to ensure that that additional growth from stem cells do not overgrow, because overgrowth of those cells could turn into a tumor and a cancer. But that is the kind of technology that's in front of us in relation to stem cells and stem cell therapy and genomics. So now the next part that's very exciting to discuss about genomics is cloning. And sometimes people don't realize genomics plays a role, or CRISPR specifically plays a role in cloning. So we're going to get into that. So right now, you literally could pay $50,000 to clone your pet or your pets. Ask Barbra Streisand. Remember Barbra Streisand? Anyway, I was going to sing a song about, anyway, I won't do that. But when you think about cloning, if a really oversimplified way to describe cloning is a DNA from a sample like skin, blood, come from a living donor, and then, or a living organism, or your dead one, could be like for the mammoth DNA, but are then inserted into a denucleated or an egg that doesn't have a nucleus, where the nucleus was removed. And so when the nucleus from the egg is removed and the DNA from a living organism is introduced, that egg then begins to reproduce with that genomic code from the donor. And that's how cloning works. So you could take a dog that died, take a sample of their DNA, take that DNA and insert it into an egg with the nucleus removed. That egg can then be, grown in a uterus of a healthy female dog, and then the dog that is born is a replica of the dog that died. So that happens today. With dogs, etc. So my son loves dinosaurs. He would love to see us clone dinosaurs, but DNA sequences from dinosaurs are not accessible. The dinosaurs died off far too long ago. The proteins from DNA have denatured. We just don't have access to that information. But we do have access to Ice Age animals like the woolly mammoth and other creatures. So there is a possibility of reviving extinct animals. What does this have to do with human health or genomics? Well, there's a very interesting research team located at Harvard that is working on using CRISPR gene engineering to copy and paste DNA from the mammoth genome into living elephant cell cultures. So remember, we have to have a living donor egg in order to clone. So you can't clone a full mammoth. We can only create a hybrid if we take mammoth DNA and insert it into the donor egg of an Asian elephant, for example. But what this research team is doing is using CRISPR to, they're essentially finding all of the fibroblast or cell culture lines that are associated with, that are unique to a mammoth genome, and mapping those, and then using CRISPR to copy and paste DNA from the mammoth, which would eventually create mammoth-like cells that would be rewritten into Asian elephant cell lines, generating over time more mammoth-like cells with high levels of precision. So, for example, you could create the mutations that are responsible for mammoth hemoglobin, and edit that into a cell line with CRISPR. The extra hair growth that we see in mammoths, what genes are associated with that? Can we CRISPR those genes into an Asian elephant cell line? So as that process continues, it's essentially possible for a mammoth to be completely recreated through gene editing. Now, where this gets even more interesting is when stem cells, induced stem cells come into play. So induced stem cells, as we discussed earlier, can be developed into multiple tissue types for studying the effects of mammoth mutations on the traits of cells, like red blood cells that carry oxygen. What oxygen carrying capacity do these cells have that are different in mammoths than there are in modern elephants? So what's next? The woolly mammoth and Asian elephant are very closely related. About they share 99.96% of their genes. So once a woolly mammoth trait has fully appeared and is sufficiently have sufficiently appeared in stem cell derivatives or stem cell derived tissues, scientists can begin experiments to generate embryos that are fully mammoth, really, possibly through stem cell embryogenesis. This would also include constructing an artificial uterus that would gestate these mammoth embryos that we've basically used CRISPR and stem cell or induced stem cells to create. And so this planned technique would eliminate the need to use Asian elephants as surrogates to breed a new generation of woolly mammoths. Once a first generation of modern woolly mammoths are born, they will be in the care of captive Asian elephant family groups. I really look forward to seeing that in zoos. So they would provide growing mammoths the social imprinting they need to form snow-treading herds in the tundra. So their adopted Asian elephant parents will not be able to tolerate cold weather for extended periods of time. So baby woolly mammoths can still be fostered in cold climates, or they would need to be, in order to survive in the wild. And then the next step would be to ensure a captive breeding population of woolly mammoths is large enough. Herds can be established at suitable grassland restoration sites throughout the Arctic. Imagine that. That is a real-life Jurassic Park, except it's an Ice Age park. So the premier future for mammoths would be something like the Pleistocene Park that has been established in northeastern Siberia. So what the hell is a Pleistocene Park, you might ask? Well, it is a Russian nature reserve in the Sakha Republic. It's located in northeastern Siberia, not Iberia. If you're Trent Sutton, it's not Iberia, it's Siberia, where they've recreated a northern subarctic steppe grassland ecosystem that flourished during the last glacial period. So this project is being led by Russian scientists Sergei Zimov and Nikita Zimov. They are testing the hypothesis that repopulating with large herbivores from the Ice Age and predators, we could restore rich grasslands and ecosystems that were caused by overhunting, because they hypothesize that a tundra is not a natural habitat, that it really should be full of grasslands, but early human hunting, Neanderthal hunting, destroyed the ecosystem, which resulted in these tundras that have permafrost, and that has actually led to a degree, and I don't understand why, but they have hypothesized that leads partially to our problems today with global warming. So by reintroducing these extremely large herbivores and carnivores from the Pleistocene era or Pleistocene period, the tundra would be reinvigorated, grasslands would grow, and we'd have some kind of efficient carbon sinking mechanism through that ecosystem. If someone knows more about that, please comment below, because I don't quite understand it. But I do want to say what's cool about this is that they are planting other extinct animals as part of their cloning effort. So that includes the woolly mammoths, cave lions, steppe bison, woolly rhinoceroses, Irish elk, and cave bear. Now, as for cloning humans, for those with conspiracy theories that love to think about science fiction in reality, there are something called spindle proteins that are in cells, mammalian cells, but in humans, those spindle proteins are located near the nucleus of reproductive cells in humans. So in order to clone a human, if you think about taking an egg and taking the nucleus out of the egg and then introducing genetic material from someone else, taking the nucleus out of it, a human egg would destroy the egg because of how close the spindle proteins are located near the nucleus. This is unique to humans and some primates. So it is not possible to clone a human, at least today. So what can we learn from today? I think one of the things that I want to impress on viewers' minds is the importance of science and scientific advancement, and to not be so fearful, but to be hopeful. And then to really understand what the data is telling us about science instead of relying on someone's Facebook post. I've said this before, but the opinion of someone you respect and care about is not evidence, it's not data, and it isn't accurate. There are ways to critically think so that you can understand what the data is telling us and then make decisions about how to live your lives and how to raise your children so that you don't become fearful of vaccines and turn into anti-vaxxers or try to cure cancer through alternative medicine that a chiropractor might offer you through essential oils or an adjustment to your back or a massage. And the list goes on, right? Rather, there are incredible advances in biotechnology that are absolutely revolutionary and will forever change our future and how we live. This will mean, ladies and gentlemen, extended lives. Our children, my children, who are ages 13 down to 6, they will probably live into their hundreds. Many of the viewers here are going to easily live into their 90s, if not early hundreds. And those long lives will probably be high quality lives lived because of our technologies in health and science. So please don't reject science and technology. Don't reject what we've learned from advances in genomic medicine, precision medicine, but rather embrace the learning process. Don't be lazy and just call up your friend or check on Facebook regarding something about medicine. Rather, educate yourself through critical thinking. Now, remember, critical thinking requires that you are willing to you are willing to challenge your own bias. So, and look at evidence. So we could do a whole other podcast on what critical thinking is and what evidence is, but that will be for another show. So thank you for joining us tonight, and please remember to like, subscribe, comment, ring the bell, and please submit more topics. We have some really exciting topics coming up relating to the real estate market, and if there's a bubble that's about to pop. And there's also an upcoming episode on polygamy. So join us, like, subscribe, comment. Peace out.